Okay, welcome to this video. In this clip, I'm going to walk you through how we solve a 3-point linkage map. Okay, and so the cross I'm going to use, the data I'm going to use, come from this cross of a 3-gene heterozygote that you see here with a homozygous recessive for those same 3 genes. So this would be a test cross. Okay, because we're crossing something with homozygous recessive. Okay, now these 3 genes, P, Q, and R, are found all on the same chromosome. And you can see that when we do this cross of this heterozygote to the homozygous recessive, we get the 8 phenotypes that are shown right here. Okay, now this seems like a lot to kind of take in, right? And there's actually more information here in these genotypes than we actually need, okay? So the first thing I'm going to do is I'm actually going to erase some information and kind of make this a little bit more easy to digest, easy to take in, okay? Now, we're looking at crossing over, generating these types, right? Some of these 8 types. And we can actually only detect crossing over in one of the parents in this cross. We can detect crossing over in the heterozygote. That's not to say that crossing over is not occurring in the homozygous recessive, the test cross parent. But because that individual is homozygous, we can't detect the results of recombination. Okay? In every case, this test cross parent is going to pass on a P, a Q, and an R allele. And for these 3 genes, the plus alleles are dominant to the no plus. Okay? So, because the test cross parent is only passing on a recessive allele for each of these 3 genes, including those recessive alleles from the test cross parent in the full genotype doesn't really add any information. It just kind of clutters everything up. Okay, so the first thing I'm going to do is I'm going to erase the genetic contribution from the test cross parent. So from each genotype in these offspring, I'm going to take out 1 P, 1 Q, and 1 R allele. That's going to leave me with just the genetic contribution from the heterozygous parent. Okay, so for example, in this first genotype that's listed, the 580, I have a recessive P that is not necessary. It's coming from the test cross parent. I have a recessive Q allele coming from the test cross parent, and I have a recessive R allele coming from the test cross parent. So erasing those test cross alleles, those alleles inherited from the test cross parent, I'm left with just the alleles that are inherited from the heterozygous parent, the P, the Q plus, and the R plus. Okay? So I'm going to do the same thing for each of these other 7 genotypes listed here. I'm erasing 1 P, 1 Q, and 1 R allele. Okay? Erasing just a recessive allele because that is what came from the test cross parent. Okay? So that's leaving me, again, as I go through this, it's leaving me with just the allele that came from the heterozygous parent. Okay, so I take out a P, I take out a Q, and I take out an R. Again, P, Q, and R, and finally take out a P, a Q, and an R. Okay, and I'll just take out this line here while I'm at it too. Okay, so now I have, for each of the categories listed in the offspring here, I have just the alleles that came, again, from this heterozygous parent. Okay. Now, if you remember back to the previous video, the first thing I need to do when I'm figuring out a 3-point test cross is I need to identify which of the offspring represent parental types, which ones represent single crossovers, and which ones represent double crossovers. Okay. Now, technically, you'd want to look at the parental generation, the crosses that generated that heterozygote. Okay. I'd want to look back at that generation to figure out which ones are the parental types, right? But I'm not given that information. So I have to use an alternate strategy. Instead, I know that the parental types are always the most common, okay? So which ones are the most common? Here, 580 and 592 are my most common types in the offspring. So those two are going to be my parental types, okay? Those are the parental types. Just really, literally the numbers are telling me which ones are the parental types. Similarly, I know the double crossovers are the least common. Okay, so that means that these bottom two, 3 and 5 here, those are going to be my double crossovers. Okay, my single crossovers are going to be intermediate in frequency. And so these 4 in the middle are going to be my single crossovers. And notice, I really, instead of having 4 types here, this is really better listed or better described as 2 pairs of types, right? 2 pairs of similar values, okay? So I have 45 and 40 are 1 pair, okay? And that is going to be a single crossover in one region. 89 and 94, similar values. Those two are going to result from single crossovers in another region. Okay, now I'm not going to worry for right now about which single crossover is happening between which pair of genes. Okay, I have identified my 4 types, and so now I want to go on to step 2, which is to figure out the order of the genes. And to do that, I only need to know which ones are the parental types and which ones are the double crossovers. Okay, the most common and the least common. Now, to figure out my orders, there's really 2 ways that you can approach this. Okay, one way takes a little bit longer than the other, but people commonly have a difficulty seeing how the shorter technique works. And so I want to talk you through both of these techniques and show you that they both arrive at the same answer. Okay, so let's do the first technique for figuring out the order of genes. Okay, what we're going to do is we're going to, if you think about 3 genes, right, there's no difference. If you have genes A, B, and C in that order on a chromosome, there's no difference between the orders ABC or CBA. They're both mirror images of each other. Okay, so what actually differentiates one order from another is which gene is in the middle. Okay, now with 3 genes, there's only 3 possibilities for which gene is in the middle, right? In this case, it could be P in the middle, it could be Q in the middle, or it could be R in the middle. Okay, so what we're going to do is we're going to take the parental types in each of those possible orders, and we're going to simulate a double crossover event and see if what we get from that order matches what we observe as double crossovers in our data. Okay, let me walk you through this. Let's just assume that the order that's given with Q in the middle is the correct order. Okay, we're going to take our parental types, our 2 parental types in that order, and we're going to simulate a double crossover. So let's take our 3 genes, P, Q, and R, okay? And in the parental type, we have P, Q plus, and R plus as one linkage unit. So there we go, P, Q plus, and R plus. The other chromosome in our heterozygous F1 would be P plus, Q, and R, okay? So these would be the parental linkage units in our heterozygous F1, okay, showing the order with Q in the middle. Okay, and we're just going to assume that that's correct. Let's see if we can justify that assumption or reject that assumption. Okay, so if we have a double crossover, then we would have one crossover event between P and Q, another crossover event simultaneously between Q and R, okay, and let's see what that would give us. So we would start at one end of the chromosome. We would have the gene P, then we cross over, we get Q, then we cross back up to the top chromosome and we get R+. Okay, so one type that we would get would be P, Q, and R+. Okay, let me use a different color here to show the other result of that double crossover. If we start at one end of the bottom chromosome now, we get P+, we cross over and we get Q+, then we cross back down and we get R. Okay, P+, Q+, and R. Okay, so are these linkage units what we see in our observed double crossover, right? Let's start with this one. P, here's P, Q, this has Q+ attached to P, right? And then R+, this has R attached to P. So what we predict from this order with Q in the middle is not what we see in our observed double crossover, so that means this is not the right order. Okay? Let's try a different order. So let's take our 3 genes. Again, I'm going to put P in the middle this time. Okay, so let's put Q on one side, P in the middle, and R on the other side. Okay, now our linkage groups that we see in the parental type have P with Q+ and R+. Okay, P with Q+ and R+. We want to keep those linkage groups intact. We're just shifting up what the order of the genes are. Okay, the other parental type with P in the middle, we would have P+ with Q and R. Okay, so there we go. Now, if we simulate a double crossover with the order of P being in the middle, then let's start at the top here. We have Q+, we cross over, we get P+, we cross back up, and we get R+. Okay, so with P in the middle, our double crossover would have the alleles in coupling, right? Q+, P+, R+. Okay, the other chromosome, if we start at the bottom here, we have Q, then we're going to cross up to the top, we get P, cross back down, and we get R. Okay, Q, P, and R. Okay, so if P is in the middle, then our double crossover, we expect to have all the alleles in coupling. Is that what we observe in our double crossovers? No, again, coming back down here, we have P, we have Q+, and R. Those alleles are not in coupling, right? So this is not the correct order. So we'd better get the right thing with R in the middle, because that's the only other possible order. Okay, so let's try this again. We'll put P, we'll put R in the middle, and then Q. Okay, remember in our parental types, our linkage units are P with Q+ and R+. Okay, so R+, Q+, and P together. Our other chromosome, P, R, Q, we have R in the middle. We need a P+ on the other chromosome with R and Q. Okay, those are the parental types, right? Now it's going to simulate a double crossover, okay, and let's see what we get. We're going to start here with P, we cross over and we get R, then we cross back up and we get Q+. Okay, P, R, and Q+ is one double crossover type, okay? The other one, if we start at the bottom here now, bottom left, we go P+, then we cross up and get R+, cross back down and get Q. P plus, R plus, and Q. Okay, so one chromosome has P and R with Q plus. Okay, is that what we see in our observed double crossovers? Right, P and R with Q plus. That is what we see. Right, the other chromosome, the other double crossover chromosome has P plus and R plus with Q. P plus and R plus with Q. Okay, so that is the correct order. R is in the middle. Okay, now that's one way to go about it. And this is as long as that could possibly take, right? So once you find the correct order, you don't necessarily need to test all three possible orders, right? If the first one you try happens to be the right order, then you don't need to test the other two. If the second order you try happens to be the right order, you don't need to test the third, okay? So that could take quite a while. That could wrap up pretty quickly. It depends on how lucky you are in arriving at the right order, okay? Another way you can do this is you can write out our parental type. Write out our two parental type chromosomes, okay? And I'm going to use the order that's given because that's probably where you would start, right? So we have P, Q plus, and R plus is one parental type. P plus, Q, and R, right? So just writing these as they're shown here. They're two parental types, okay? So we want to compare that visually to our double crossover types as they're given in the list, right? P, Q plus, and R, okay? P plus, Q, and R plus, as we see here. Okay, now let's take a look at this. We're going to compare one parental type chromosome to another parental type chromosome, okay? And I would just start, work your way left to right, and you could match up, directly compare the two top chromosomes or the two bottom or one bottom to one top. I would just start with like what's looking similar, right? So here we have a P there. That is linked to a Q plus. That's linked to a Q plus here. And that is still linked to an R plus on the parental type. It's linked to an R in the double crossover. Okay, so in this linkage unit that we're looking at, we have P linked to Q plus linked to R plus in the parental type. In the double crossover, P linked to Q plus linked to R, right? So R is different. How that is arranged on the chromosome between the parental type and the double crossover. So that different one is the one that's in the middle. Okay? If we compare these two bottom chromosomes, right? P plus, Q, Q, R versus R plus. R is different, so R is in the middle. Okay? And so that is our second method for determining the order. Okay, so having determined the order, we can start to make our linkage map. Okay, so there's our chromosome. There's our 3 genes on the chromosome. I'm going to write my 3 genes in here in the correct order. P, R in the middle, and Q. If you chose to, you could write this left to right Q, R, P. That's fine. Okay, what's important is that R is in the middle. That makes that a unique order. Okay, now what I'm going to do is I'm going to make some space for myself. We figured out the order. Hopefully you took some notes on that. So I'm going to erase that work and make some room for myself to figure out the distance between our genes. Okay? Now, in our map here, we have one region. I'm going to call this region 1. This is from our first gene to our second gene, P to R in this case. Okay. Our region 2 is from the second gene to the third gene, R to Q in this map. Okay, so P to R is one distance that I need to figure out. R to Q is another distance that I need to figure out. The third distance is from one outside gene to the other, from P to Q, this entire distance. Okay, and as we saw in that video about additive maps, the P to R distance plus the R to Q distance should equal the P to Q distance. Okay? So, what I need to do to figure out distances between these pairs of genes is I'm going to add up all of the recombination events that happen in region 1 in order to get this distance from P to R. Okay? Now, I have single crossovers. One pair of my single crossovers represent crossovers between P and R. So before I start doing my calculations to get the distances, I need to know which single crossovers happen in region 1 and which single crossovers happen in region 2. Okay, and to do this really quick, I'm going to go back to the same thing I did to figure out the order. Okay, I'm going to take my parental types, and remember that the way they're written here is in the wrong order now. Okay, so I want to make sure that I write my genes in the correct order. P, R, Q. Okay, I'm going to take my parental types in the right order, right? So P, Q plus, and R plus is one linkage group. The other parental type linkage group is P plus with Q and R, right? Okay, so these are my parental types in the correct order with R in the middle. What do I get when I have a single crossover in region 1 between P and R? Okay, well, let's look. Let's start at this top chromosome. We get P, crossover, we get R, and we stay on that bottom chromosome with Q. Okay, so P, R, and Q, the alleles in coupling. Okay, the other chromosome from this single crossover, I start at the bottom left here. I get P plus, crossover, I get R plus, and then Q plus. P plus, R plus, and Q plus. Okay, where do I see those single crossover types in coupling? That is right here. Okay, so these two, 89 and 94, represent a single crossover between P to R. Okay, now, what if I can just verify for the other two? I have P, R plus, and Q plus are my one parental type. P plus, R, and Q are my other parental type. If I have a single crossover in region 2 between R and Q, I'd better get these two types, right? Let's just verify that. I start here at the upper left. I get P, stay on that chromosome. I get R plus, then I cross over and I get Q. Okay, so that gives me P, R plus, and Q. Okay, same thing that we see up here, P, R plus, and Q. Okay, the other chromosome, I'd start on the bottom left. I get P plus, I get R, then I cross over and I get Q plus. P plus, R, and Q plus. Okay, over here, P plus, R, and Q plus. So yes, these two with 45 and 40 as my counts represent a single crossover between R and Q. Okay, so now, finally, I figured out which types are what. I figured out which single crossovers are happening where. I figured out my gene order. Now I can calculate distances. Okay, so again, let's make a little bit of room here to do that. okay? And so I'm going to start on the left here. Let's do the P to R distance, okay? Now, like I said, I want to add up all of the recombination events. This is going to be in the numerator, okay? All of the recombination events in that region, right? So my single crossovers between P and R, these two types, I need to add in there, 89 plus 94. I also have a crossing over event between P and R represented in all of my double crossovers. Okay, so I need to add those in too. I'm adding up recombination events, okay, in that specific region. I am going to divide by my total count, okay, and I'm going to multiply this by 100 so that I'm expressing this as a percent, okay? And that comes out to be 13.2 centimorgans. Okay, so that's my distance, 13.2. Okay. Now, my other distance that I need to figure out, my next distance, I should say, is between R and Q. Okay, and again, I'm adding up all of the recombination events that happen between R and Q. Okay, so these two single crossovers, 45 and 40, have a crossing over event between R and Q, but my double crossovers also have a crossing over event between R and Q, so I need to add them in again. Okay, divide by my total, 1448, and multiply by 100 to express as a percent. Okay, and that comes out to be 6.4 centimorgans. Okay, so that is 6.4 there. So to get the distance, the third distance between P and Q, okay, I could just rely on the fact that the map is additive and take 13.2 plus 6.4, okay? However, if I did either of these calculations incorrectly, then I'm never going to know it. Okay, on the other hand, if I calculate the P to Q distance from my data independently, then if I did a math error up here or up here, I could catch it. Okay, so what I'm going to do is I'm going to, again, between P and Q, I'm going to add up all of my recombination events. Okay, so the single crossovers, all four of them, all represent one crossover event between P and Q somewhere. Okay, so let's take 45 plus 40 plus 89 plus 94. Okay, those are my four single crossover types. The double crossovers had two crossover events in this whole region, each one of them. Okay, so I'm going to multiply my 3 plus 5 double crossover events, multiply that twice, add that in twice, once for each of the crossing over events that they represent. Okay, divide this by my total and multiply by 100 to represent as a percent. And that comes out to be 19.6 centimorgans. Okay, so 19.6 is my distance there from P to Q, and you can see that 19.6 equals the sum of 13.2 plus 6.4. So my map is additive. I did all my math correctly. Okay, so through these steps, I have arrived at my linkage map, and that is the answer to this problem. Okay, the 3 genes on the chromosome in the correct order with all pairwise distances represented.